Masking-based methods learn by prediction. Fill in the blank. Contrastive methods learn by comparison. Know what's similar and what's different. Both require no labels. Both produce powerful representations. The contrastive learning recipe. Take an image. Create two augmented views, crop differently, flip, adjust color, blur slightly. These two views are a positive pair. They show the same content from different perspectives. All other images in the batch are negative pairs, different content. Train the model to produce embeddings where positive pairs are close together and negative pairs are far apart. The loss function bring the two views of the cat together in embedding space, push the cat away from the dog, the car, and the landscape. The model learns to recognize content despite superficial changes in appearance. SimCLR from Chen et al. Uses the infant's contrastive loss, a softmax over cosine similarities in the batch. It requires large batch sizes, 4,000 or more, to provide enough negative examples. BYOL eliminates negatives entirely. A student network learns to predict the output of a slowly updating teacher network, applied to a different view of the same image. DINO combines self-distillation with vision transformers and discovers semantic segmentation as an emergent property. The attention maps naturally highlight object boundaries. For histopathology, DINO-style models learn to segment tissue regions, identify cell types, and detect structural abnormalities from unlabeled slide images. Contrastive learning adapts naturally to biology. CellContrast augments gene expression profiles, masking random genes, adding expression noise to create positive pairs from the same cell. Cells of the same type cluster in the learned embedding. Clip-style multimodal learning pairs protein sequences with text descriptions of their function. The embedding learns to align sequence and language, enabling text-based protein retrieval. Multi-omics contrastive learning, RNA-seq and ATAC-seq profiles from the same cell are a natural positive pair. They're two views of the same cell state. The contrastive objective learns a joint representation that captures information from both modalities. Beyond masking and contrastive learning, other pretext tasks force models to learn specific representations. Rotation prediction: rotate an image by 0, 90, 180 or 270 degrees and predict the angle. The model must learn object structure to tell up from down. Jigsaw: shuffle image patches and predict the original arrangement. The model must learn spatial relationships. Colorization: given a grayscale image, predict the colors. The model must learn what objects look like. Each pretext task biases the learned representation differently. Choose the task that requires the features most relevant to your downstream application. How do you evaluate self-supervised representations without labels? Linear probing: freeze the entire pre-trained backbone, attach a single linear classification layer on top and train only that layer with a small label dataset. If the linear classifier achieves high accuracy, the frozen representations already separate the classes. They're good representations. Key nearest neighbor evaluation: classify each test example by the majority label among its k nearest neighbors in embedding space. No training at all, purely geometric evaluation of the embedding quality. Both methods measure how well the self-supervised task induced useful biological structure in the representation space. Mask and predict next token, masked autoencoders. Augment and contrast: SimCLR, BYOL, DINO. Pretext tasks: rotation, jigsaw, colorization. The data labels itself: 250 million protein sequences, 30 million cells, millions of tissue images, all free training data. Self-supervised learning turns this abundance into powerful representations. It's the engine behind every foundation model in the polyglot video.